Welcome to Biology at Priyojit, my dear viewers. Today we'll explore enzymes, the tiny but powerful biological catalysts that drive every chemical reaction inside the living cells. From their nomenclature and systematic classification to their specificity and mechanism of action, enzymes ensure that life processes occur very rapidly, accurately, and efficiently. Let us understand these fascinating molecular machines in a simple, clear, and exam-oriented way. So, to introduce the concept of enzyme, we know that life always depends on two fundamental abilities. Number one is self-replication of the living organism, and second one is the efficient and selective catalysis of chemical reactions. So, enzymes are basically biological catalysts that increase the rate of biochemical reactions without undergoing any permanent change. So, the term enzyme was introduced by Frederick W. Kuhne in 1878 for substances earlier known as ferments. Eduard Buchner, a German biochemist, isolated the first enzyme and proved that enzymes can act outside living cells. He was awarded the Nobel Prize in 1907. So the protein nature of enzymes was demonstrated by James B. Sumner in 1926 by crystallizing the enzyme urease. So for this discovery, Sumner received the Nobel Prize in 1946. So this is the image of Frederick William Kuhne. He is Edward Buchner and he is James Sumner. Nomenclature of enzymes. Enzymes are generally named by adding the suffix "ase" to the substrate name, for example urease or lactase, or to the type of reaction they catalyze like oxidase, dehydrogenase, etc. So some enzymes have more than one name and sometimes different enzymes share the same name, creating confusion. So to avoid this ambiguity, an international system of enzyme nomenclature was developed, which is Enzyme Commission Classification or EC Commission Classification. Enzymes are classified based on the type of reaction they catalyze. Each enzyme is assigned a unique Enzyme Commission number or EC number with 4 parts like example. So for example EC 2.7.1.1. so meaning of EC number, that is first number is the main class, that is it can be transferase or oxidoreductase or hydrolases or lyases or isomerase or ligases. The second number always include the subclass and the third number indicate the subclass, and the fourth number is the serial number of the enzyme in that subclass. So the complete list of enzymes is maintained by the Nomenclature Committee of the International Union of Biochemistry and Molecular Biology, which is IUBMB. Classification of enzymes. There are 6 major classes. First one is the oxidoreductase, catalyze oxidation reduction reactions. It involves transfer of electrons, hydrogen atoms or hydride ions. Examples include oxidases, dehydrogenases, oxygenase and peroxidases. So here alcohol dehydrogenase, it acts on ethanol to produce acetaldehyde. Transferases, it catalyzes group transfer reaction from one molecule to another. Transferred groups include methyl, amino, glycosyl or phosphoryl groups. Examples include kinases, phosphorylases, transaminases and transcarbamylases. So this is transferase enzyme. They add ATP with the substrate and it will phosphorylate the substrate and one molecule of ADP will be formed. Hydrolases, they can catalyze hydrolytic cleavage of bonds using water, acts on CC, carbon-carbon, carbon-oxygen, carbon-nitrogen and phosphorus-oxygen and other bonds. Examples include phosphatases, phosphodiesterase, peptidases, etc. This is the example image showing the reaction of compound with the H2O and it will form the AOH and BH. Lyases, it catalyzes bond cleavage without hydrolysis or ATP. It results in the formation of double bonds or addition of groups to double bonds. Examples included aldoses, aldolases, decarboxylases, dehydratase and synthesis. So the pyruvate decarboxylase acts on pyruvate to form acetaldehyde and ultimately ethanol will be produced. Isomerase, it catalyze rearrangement of atoms within a molecule. That is, the isomerase converts D-glucose into D-galactose. They convert one isomer to the another. So examples include mutase, racemase, cis-trans isomerase, etc. Ligases or synthesis, they catalyze joining of two molecules. This is the joining of A and B with the help of a XTP molecule. The reaction is coupled with ATP hydrolysis or CTP, UTP, GTP hydrolysis, formation of CC, CO, CN or CS bonds. Examples include synthetase, carboxylases, etc. Unique properties of enzymes. These are the unique properties of enzymes. Enzymes possess certain exclusive properties that make them highly efficient molecular devices. Chemical nature of enzymes, except for a small group of catalytic RNA molecules or ribozymes, all enzymes are proteins. Enzyme activity depends on the native or three-dimensional conformation of the protein. Denaturation or dissociation into subunits usually results in loss of catalytic activity. Breakdown of enzymes into individual amino acids completely destroy their activity. So therefore, all levels of protein structure are very much essential, primary, secondary, tertiary and quaternary structures. Molecular structure of enzymes. Enzymes vary widely in molecular weight, ranging from 12,000 to 1 million daltons. Based on composition, enzymes are of basically two types: simple enzymes composed only of amino acids, and conjugated enzymes contain a non-protein component. A hollow enzyme, that is a complete catalytically active enzyme, consists of both apoenzyme or apoprotein, that is the protein part, or cofactor, coenzyme and prosthetic group, that is the non-protein part. Specificity of the enzyme. We know enzymes are very much, they are highly specific in nature. Each enzyme acts on a particular substrate or a closely related group of substrates. This specificity ensures high efficiency, accurate biochemical regulation. Mode of enzyme action. Enzymes increase the rate of reaction by lowering the activation energy. They do not alter the equilibrium position of a reaction. Enzymes basically change the speed at which equilibrium is reached, but not the final outcome. So what is the non-protein components of the enzymes? First one is a coenzyme. Coenzymes are small organic recyclable molecules. They act as group transfer agents or carriers. They stabilize unstable groups like hydrogen atoms or hydride ions. They transport groups include methyl group like folates, acyl groups like coenzyme A, and oligosaccharides like dolicol. Prosthetic group, non-protein components when tightly bound with the enzyme, that is prosthetic group, and then this binding can be covalent or non-covalent. Examples include pyridoxal phosphate, flavin mononucleotide or flavin adenine dinucleotide, thiamine pyrophosphate, biotin, or any other metal ions like cobalt, copper, magnesium, manganese, selenium and zinc. Enzymes containing tightly bound metals are called metalloenzymes. About one third of all enzymes are metalloenzymes. Cofactors that bind loosely and transiently to enzymes or substrates. They must be present in the reaction medium for enzyme activity. They are mostly metal ions. Enzymes requiring such ions are called metal activated enzymes. Differences include metalloenzymes where metal is tightly bound, and metal activated enzyme, that is metal is loosely bound. This is the cofactor which bounds with the inactive protein and make it after binding with the enzyme or protein, cofactor make it active. Isoenzymes, multiple molecular forms of an enzyme that catalyze the same reaction are the isoenzymes or isozymes. They are found in the same species, in the same tissue, even in the same cell, but they differ in their kinetic properties, regulatory behavior, cofactor performances, that is like NADH versus NADPH, cellular location that can be soluble or membrane bound. Isoenzymes have similar but are not identical amino acid sequences. Their common evolutionary origin. Examples include lactate dehydrogenase or LDH. Exist as 5 isoenzymes in vertebrates, and they are separable by means of electrophoresis. Mechanism of enzyme action. Enzymes are active only in their native 3D conformation. Each enzyme has a specific region called the active site. The molecule that binds to the active site is called the substrate. The active site contains amino acids that bind and catalyze reactions. Active site regions, binding site that binds the substrate. Catalytic site carries out the reaction. Common amino acids at the active site like tyrosine. These amino acids like tyrosine, histidine, cysteine, glutamic acid, aspartic acid, lysine and serine, they are found in the active site of the amine enzyme. Enzyme substrate complex, when this substrate is formed, the concept was proposed by Michaelis and Menton. Enzyme and substrate combine to form enzyme substrate complex, and the enzyme releases the product and remains unchanged. So the reaction sequence must be E plus S. It will form the ES complex and then it will break down to produce E plus P. ES complex is also called as Michaelis-Menten complex. So this is the image showing the ES complex formation and the formation of product from the enzyme substrate complex. Catalytic strategies of enzyme. First one is the covalent catalysis. Temporary covalent bond is formed between enzyme and substrate. Enzyme has nucleophilic or electrophilic groups. Examples include chymotrypsin. This is the image showing the covalent catalysis of enzyme. In general acid-base catalysis, active site amino acids act as proton donors or acceptors and they enhance reaction rate. Examples include lysozyme. So this is the image showing the general acid-base catalysis performed by enzyme. And metal ion catalysis, also called as Lewis acid-base catalysis. Metal ions assist in redox reactions and stabilize negative charges. Example include cytochrome C. So this is a metal ion catalysis reaction. Models of enzyme substrate interaction. Lock and key model. Here we know that active site is rigid and preformed. Substrate fits exactly like a key in a lock. It explains high substrate specificity of the enzyme. So lock and key model is basically a model that used to represent enzyme substrate interaction. Now, the second model is the induced-fit model, which was proposed by Coastline. Here, active site is flexible, not rigid like the lock and key concept. Binding and of substrate induces conformational changes in the enzyme, and its advantages: it explains broad specificity like lipase. It explains mechanism of catalysis by stressing substrate bonds. So, the induced-fit model is a model depicting the enzyme-substrate interaction. The enzyme and the substrate undergo structural changes to attain an optimal fit. Activation energy and catalysis. Catalysts increase reaction rate without affecting equilibrium. Ground state, energy level of substrate or product molecules. Transition state, that is the active region, active state, and this is the ground state. This is the transition state. Highest energy point, transition state has the highest energy point, equally likely to form the substrate or product, and it is highly unstable, so it will from in the ES to it will give the product. Activation energy, this is the activation energy or EA, energy difference between the ground state and transition state. Enzyme enhance reaction rate by lowering activation energy, not by changing the delta zero. So my dear students, we have seen how enzymes act as highly efficient biological catalysts that regulate every metabolic reaction in living cells. Their systematic nomenclature and classification help us understand enzyme diversity, while the mechanism of action explains how reactions occur with speed and precision. So keep revising, stay curious, and let enzymes reveal the chemistry of life to you. Thank you and see you in the next class. Bye.